This is a production of Cornell University.
I'm speaking to you today though from my home and roughly where that yellow star is, where I work in an urban and regional food systems lab. This means that the farmers that we work with are very diverse in their scope and practices, but our research broadly supports farmers that are serving small-scale regional markets across the upper Midwest. I also wanted to mention here that Wisconsin is also home to the 2 working public carrot breeders in the state, in the nation rather, and we're really lucky to be able to work with them on this project as well. So in order to meet the needs of the farmers we work with who value things like flavor and weed competitiveness as well as open pollinated varieties, we're interested in being able to use germplasm from the USD carrot collection in order to introgress traits that are of interest to us. The carrots in this collection are relatively unstructured and carrot itself is an outcrossing diploid. We are also hoping that the approach that we're taking can serve as kind of a case study or a proof of concept approach for other groups that are interested in using germplasm collections in breeding, but need methods to be able to do so deliberately and efficiently in order to be successful, whatever the size of their program. Before I begin to talk about the approach that we're using, I did want to touch just briefly on one of the traits that we're interested in a little bit more in depth, because I'll be referencing it again throughout the talk. And so canopy height is simply the measurement between the soil and the top of the longest petiole. And we're interested in this because it's positively correlated with weed competitiveness, which is something that's really important to a lot of our growers, many of whom are organic and spend a lot of time and effort, especially earlier in the season, managing weeds in their field. This is a phenotype we're able to easily measure. So we have methods both for whole plot phenotyping in the field, and then using barcode rulers and scanners, we can quickly do single plant phenotyping in the lab as well. And now I'm going to discuss a little bit about our approach. And we're interested in using genomic prediction and broadly genomic assisted selection in order to quickly identify useful germplasm from a collection while minimizing the need for extensive field screening. And with this work, we're really drawing on 2 sources of inspiration. The first is that of core collections. Now, usually these were, these are methods used that have been used to reduce the size of collections that need to be screened in the field. Typically, they've been based in phenotypes or passport information. With the increasing accessibility of low-cost markers, however, many germplasm curators are able to genotype the entire collections, making them amenable to things like genomic prediction. Over the last 5 years or so, there's been an increased interest in using genomic prediction in germplasm collections, but most of these studies have been based in simulations. So what we really want to do is directly compare phenotypic and genomic assisted selection by developing breeding populations using different methods and then taking them into the field. So just really briefly, one slide on the prediction models we're using. These are kinship-based methods in which we're building models by regressing phenotypes on markers in order to calculate breeding values of untested accessions. And here's our experimental design. We are comparing 3 different selection strategies, 2 different flavors of genomic assisted selection against phenotypic selection. And then for each of these, we're taking 5 highly ranked accessions for each strategy trait combination. You'll see harsh flavors on the screen where I don't have time to talk about that today, but it's another thing that we're really interested in. And each of these accessions is being crossed to 2 popular inbreds, which can be thought of as either biological replicates or kind of like a tester. And then these are progressed to the F2 generation. So the phenotypic selection is serving as kind of our current standard approach using data from Hancock, Wisconsin a couple years ago. The first flavor of the genomic selection, we're terming the genomic breeding values strategy, in which we're using available breeding program data to calculate breeding values for untested accessions. I know I'm kind of racing through these. I'm more than happy to talk more in detail about these strategies later in the chat. And then finally, we are also working with the genomic training population strategy, in which we identified a genetically representative training population, which we then phenotyped in our target environment. This one maximized the relationship between the test and the training population, which can dramatically increase predictive accuracy of models, but it did require an extra year of phenotyping before breeding could begin. Next, I'm going to talk about just some of the results we've been able to achieve so far. Obviously, the main thrust of this work is going to be in the comparison of breeding populations, and I'm really glad to report we just got approval to actually have a field season this year, which wasn't clear a few weeks ago. So while we have some pilot data, we're going to be able to dramatically increase that next year by doing a replicated trial of these breeding populations and measuring them for the traits we're interested in. As we were developing our genomic training population strategy, we also were able to test different parameters of the training population using cross-validation. So what we looked at was both composition and also relative size compared to the total collection, which is what you see here. And then finally, and this has become somewhat of a pet project of mine, we're beginning to analyze intra-accession diversity. Observationally, we note a fair degree of heterogeneity within a given accession. And I think moving forward, the success of this type of approach is going to rest not only on improving strategies to exploit variation between accessions, but also methods to quantify and track diversity within accessions as well. So in closing, we believe that genomic-assisted selection can help small programs use genetic resources more effectively. But what we're finding is that high prediction accuracy seems to require large training populations. But even moderate accuracy still may be useful to programs who really just need to reduce the number of accessions that they're screening. And our future work will compare phenotypic and genotypic selection in more detail, and we'll continue to track inter-accession diversity. With that, I'd like to thank people who have really been invaluable to this work, and I can take any questions. Thank you.
Great, thank you so much, Keo. I guess we'll be taking questions. There's nothing in the chat, so if you want to go ahead and raise your hand and you can ask the question. Looks like Hannah Jeffrey has a question. You can go ahead and unmute yourself, Hannah.
Thank you. My question is, about how large do you think your training populations will need to be in the future? And what kind of statistical modeling have you implemented to try to predict what size it will have to be?
Sure, thank you for that question.
No problem.
Yeah. Can you still hear me? I'm not sure what I'm looking at right now. Okay, thank you. Yeah, so we saw a pretty dramatic improvement in prediction accuracy when we increased the size of our training populations relative to the total size of the collection. So I'm seeing another question here, how many germplasm do you work with? And we were using about 470 cultivated accessions within the germplasm collection. And then the training population that we phenotyped was 10% of that size, which is consistent with previous literature that looked at core collections that were roughly 10% the size of the total collection. I think what's interesting is how dramatically accuracy did increase when we increased the relative size. So jumping from 10 to 20% had a large effect on prediction accuracy. And then the second part of your question is, how did we look at that?
That is correct, yes.
Yeah. So the one graph in the results section that I showed showed how we had a data set of accessions for which we had data for, and we used cross-validation to mask a percentage of that served as our test population, and then used the remaining as the training population in order to predict the test group. And we were able to do that many times with many different training populations of a given size calculated in the same way in order to calculate the mean and variance of accuracy.
Thank you very much. It's helpful for me as a student.
Sure, yeah.
I guess you answered how many germplasm. So Mark Sorrells, would you like to unmute yourself and ask a question?
Yeah, thanks for your talk. Aren't your phenotypes confounded by the percent heterozygosity in individual plants?
The shorter answer is probably. Right now we're working with single genotypes per accession, and this is an outcrossing species, so there's a degree of heterozygosity within accessions. And what I found with some preliminary analysis is that level of heterozygosity also varies across accessions. So I think future work, it's going to be really important to follow up with some of that and be able to account for percent heterozygosity in our predictions.
This has been a production of Cornell University, on the web at cornell.edu.